Good afternoon. The work I'm going to present involves the 3D in vitro modeling of the functional unit of the exocrine pancreas by tomographic volumetric bioprinting. This unit is the pancreatic gland responsible for the secretion of the digestive enzymes and the region where the first lesions of pancreatic ductal adenocarcinoma or pancreatic cancer develop. It is constituted by epithelial cells, acinar and ductal cells, surrounded by the pancreatic stellate cells that are stromal cells and are normally in a quiescent state. However, as a result of inflammatory stimuli, they become active and change to a spindle-shaped, exhibiting a myofibroblast-like phenotype. Once activated, these cells start to produce cytokines such as the interleukin-6, which induce in the epithelial cells the mutation of oncogenes, mainly the KRAS oncogene, leading to the development of pancreatic cancer through different neoplastic stages. The crosstalk between cancer cells and stromal cells is recognized as a key player in the tumor evolution, promoting the pancreatic cancer progression. And the complex mechanisms involved in this crosstalk, as well as the unique bioarchitecture of the PDAC microenvironment, lead to consider pancreatic cancer as one of the most aggressive diseases. Indeed, pancreatic cancer represents one of the leading causes of cancer-related death worldwide, with the lowest survival rate of all cancers. And the rapid tumor evolution without clear symptoms results in a late diagnosis and in a poor clinical prognosis. Therefore, to enable an earlier detection, we need to understand how pancreatic cancer develops from the very early stages in the pancreatic gland. Our research is focusing on the development of functional in vitro models that can shed light on this pathology and improve consequently the diagnosis and treatment. Indeed, as we all know, the in vitro models represent important alternatives to the in vivo models that are not representative of the human physiology and genetics, and have limitations such as the high cost, the low throughput processes, and the ethical issues. On the contrary, the in vitro models allow cheaper and faster drug screenings, the use of human cells, tunable cellular assembly, and provide controlled biophysical cues. So the aim of this work is to reproduce the crosstalk between epithelial cancer cells and stromal cells in a 3D in vitro model that can also mimic the complex microanatomy of the human pancreatic gland. And we use human fibroblasts embedded in a gelatin methacrylate hydrogel to mimic the stromal component, and human pancreatic ductal epithelial cells to resemble the epithelial tissue. The gelatin methacrylate solution was prepared at 5% weight volume concentration with a very low amount of photoinitiator. And then the hydrogel was characterized in terms of photoreology and NMR spectroscopy. In particular, the synthesis repeatability was demonstrated by comparing the spectra of two different batches. This biomaterial is widely used, has been widely used in our group at Politecnico di Torino. For instance, in the context of the IPRO project, the ability of this bioink in supporting the growth and proliferation of fibroblasts over several days was assessed. If you are interested in this work, you can find more details in the poster, which will be presented right after this talk, and the number is 92. In this work, we used tomographic volumetric bioprinting to reproduce such a glandular shape. So this technique consists in illuminating a cell-laden photosensitive hydrogel with visible light, and by using a sequence of tomographic back projections of the desired object. leading to the photopolymerization of the material. This technique permits to print hollow structures without the need for support and in a very short building time. Moreover, the volumetric bioprinting represents a self-friendly procedure, as the one-step manufacturing process reduces the stress experienced by cells. So here we loaded the human fibroblasts in the gelatin methacrylate solution containing the photoinitiator, and then the volumetric bioprinting was employed to obtain this 3D echinodactyl structure. In particular, a set of tomographic light patterns was projected to the vial, and the light scattering effect caused by the presence of cells inside the gel provoked a loss of resolution, such as a resulting incomplete acinus. However, by performing a numerical correction of the light dose, we obtain improvements in shape fidelity and resolution of the printed structures. The viability of fibroblasts inside the bioprinted construct was assessed by observing the state of the cells, and these live-ended images show that most cells survived after the biofabrication process, and they were homogeneously distributed in the gel after 24 hours. The proliferation and growth occurred as confirmed by the metabolic activity, which increased with time. And in particular, we observed a marked significant increase between 72 and 96 hours after printing, and this is probably due to the cell recovery after from the stress that the biofabrication may cause. These fluorescence images also show the human fibroblasts assuming an elongated shape within the gel. The ability of the epithelial cell in covering the inner walls of the bioprinting construct was also analyzed. And these microscopy images from alive and dead cells show that the HPD cells covered larger extents of the inner walls of the duct. However, the total coverage didn't occur during this experiment duration. And when co-cultured with fibroblasts, epithelial cells appear as grape-like clusters and form large linings at the gel cavity interface. The effect of the crosstalk between epithelial and stromal cells was also studied by evaluating the cytoskeleton changes in the cytoskeleton composition of fibroblasts. In particular, we see the healthy epithelial cells and epithelial cells overexpressing the KRAS oncogene within the fibroblast-laden constructs. And then thin slices of the gels were imaged upon immunostaining to evaluate the expression of the alpha-smooth muscle actin in fibroblasts. Indeed, alpha-SMA is known to be overexpressed in inflamed tumor-associated fibroblasts. Qualitatively, we observed that the cells, the fibroblasts co-culture with the HPD KRAS cells exhibited a stronger expression of alpha-SMA, which increased with time. And this result was confirmed also by a quantitative analysis showing that the expression resulted significantly higher in fibroblast co-culture with the HPD KRAS cells for 72 hours compared to 24 hours. We also studied the dependence of the alpha-SMA expression on the distance from the duct at the single cell level. And these graphs show the measurements indicating that fibroblasts closer to the ducts exhibit a higher expression of alpha-SMA. And this indicates that fibroblasts feel the effect of the HPD KRAS cells that actually laid within the ducts. In contrast, fibroblasts co-culture with healthy epithelial cells or without epithelial cells didn't exhibit a decaying degree of activation with the distance from the duct. So in conclusion, this 3D in vitro model allows to recapitulate the complex microanatomy of the human pancreatic gland and allow to study the mechanisms implicating during the initial stages of pancreatic cancer progression. And therefore, it represents a powerful tool to reproduce the stromal cells activation that occurs in vivo, study the pathology and perform efficient drug screenings, and finally elaborate personalized therapies to fight pancreatic cancer. I would like to thank all the co-workers, Professor Gianluca Ciardelli, Professor Tondaturo, people from EPFL, and of course you all for the attention. Thank you.
Thank you again for sharing this exciting work. Are there questions?
Hello, thank you very much for your talk. I was wondering if in the future it would be possible to substitute GLMA with more elaborate matrix, because the pancreatic cancer is known for lots of presence of collagens and hyaluronan who are actively participating in the activation of stellate cells as well.
Yeah, thank you for this great question. There are also other, there is, as probably you know, the collagen methacrylate material, and this is a more biomimetic material, of course. However, there are disadvantages. It's very expensive, for instance, but GELMA is recognized as a good substitute for stromal material, but definitely we can explore also other possibilities.
Thank you for your nice talk. Did you check if the fibroblasts you inserted in your model express IL-6? Because it seems like you're modeling the pathological state. Are you able to model also the healthy state where you have a not inflamed stromal port system?
Yeah, you mean if we can model like the release of cytokines or other mechanisms?
It seems like you have already this inflamed interleukin-6 contributing system. So could you model the healthy?
Yeah, actually, during my PhD, I developed other in vitro models for the study of pancreatic cancer, and there I evaluated also the release of cytokines, the interleukin-6, and actually we observed that the human fibroblasts can release the interleukin-6 when co-cultured with HPD KRAS cells. So this is something we can do and we will do.
And before you cultivate them with the KRAS mutation cells, they do not?
Exactly.
Okay.
So yeah, we compare all the conditions.
Hello, thank you very much for this nice talk. We are wondering, how did you get your HPD KRAS cells?
It's a cell line actually. So they were provided by another lab. But this is a cell line.
Thank you.
Thank you very much for the nice talk. So many cancer scientists are interested in studying how cells transition from one state to another, from a healthy state to a cancerous state to a metastasis state. And those cells will remodel their microenvironment depending on the state that they're in. As an example, cancerous cells tend to have a stiffer matrix than healthy cells. So there have been discussions where I'm from at the NIH, and I'm sure outside of how to deploy what some people call smart materials or programmable materials or living materials to study how cells transition from one state to another. So these are materials that can change their biophysical properties not only in response to user input, but in response to cell input as well, in order to create a more biomimetic environment that can be used for drug screening or to probe the mechanisms that cause these transitions between states. How do you see these kind of materials being used in your own research?
Thank you for the very interesting question. If I understood, you ask if we can somehow control the transition between the different stages, right? Actually, so now we are currently work with different cell lines, so cancer cell lines. We are using the HPD KRAS cells to like mimic the initial stage, and also other the MIA-PACA to mimic the later stage. But yes, this is something we actually want to do. So for instance, one of the main goal we want to achieve is to monitor also like the stiffening you mentioned, and so at the same time monitoring how the cancer cells migrate or how they distribute within the gene. So we are now at a very preliminary phase, but yeah, this is very interesting, and we will look into it for sure. Thank you.
We have time for a final question.
Hi, thanks for your presentation. I'm just wondering, when you make the duct, presumably there was gel and fibroblast there. What's happening? Is it being compressed? And the cells that were there, do they die? Are they pushed into the gel? Because I was just wondering, are you at risk of having a sort of a fibrotic layer underneath?
Yeah, thank you. This is a very good point. So we just try to see the epithelial cells manually, but something we want to try to make the coverage more homogeneous. We want to try a tilting movement during the seeding to distribute them, because actually they are alive, but they are more at the bottom. So yeah, this is something we want to improve, definitely.
Thank you very much, Viola, again.
Thank you.
And with this, we are at the end of this session on biomaterials and MPS. Thank you, everyone. Thanks a lot to the great presentations and the exciting work that we listened to today. And I hope you enjoy the rest of the conference, the coffee, and whatever is still coming. Thank you.